I'm back, and I'm here for the second lecture of the second part of the Bio 105 Human Biology course, and that makes it lecture number 12. And the name of what I'm going to be talking about today is Gametogenesis, which literally means the formation of gametes. Gametes are the special cells, the sperm and the eggs that eukaryotes use in order to do sexual reproduction. So I'm going to talk about sexual reproduction. And it's important that we understand these cells separately from all the other types of cells because they are genetically unique. We've already seen that they have less chromosomes in them, so we need to understand how they form, very much like we looked at the way that somatic cells form in terms of mitosis, and now we're going to learn a different process, which is how gametic cells form, and that's called meiosis. Sorry that it sounds like just the same, but we need to distinguish those. I have only one funny for you today, but I thought it was pretty good. It's a picture of an eel for some reason, I don't know why, but it says, "Why was the gamete so rich?" A gamete, remember, is a sex cell, and because sex sells is the answer to that riddle, and that's funny. Plus, it's a moray eel. I have no idea why, but that seems funny too. Anyway, let's get started. Okay, so before I get to the diagrams and all the terminology that might slow us down a little bit, I thought I would start by talking a little bit about reproduction in general. Reproduction is, we know, something that's required to define what is alive and what's not alive. Things that are alive have to be able to reproduce. Cells are alive, so cells need to be able to reproduce, and last time we talked about mitosis, which is cellular reproduction. But that is a form of reproduction that doesn't involve gametes. That's why we're distinguishing mitosis and meiosis. So I want to make sure that you know that we can stretch that not just at the cellular level, but up sometimes to some macroorganisms. And so we need to divide reproduction into two different types. The first type is asexual reproduction. This only involves one parent, just like mitosis only involved one cell which divided into two. So it only involves one parent. It produces offspring that are genetically identical, just like the two cells in mitosis were genetically identical. And it's mostly exhibited by single-celled organisms, and it's occurring by mitosis. But, like I said, there are some multicellular organisms, macroorganisms that we can see with no problem that are capable of this asexual reproduction. It's fairly simple for you to demonstrate it. If you go over to your neighbor's house and say, "I really like this house plant you have, can I take a cutting of it?" and you take a little piece of it and you maybe put some root growing hormone on it, stick it in a pot, it will grow roots and grow into a plant, and then you will have two plants where there was once one. That's asexual reproduction. It did not involve pollen or seeds or anything like that. So there's no gametes involved, and it's happening just by mitosis. The cutting that you took just starts growing new cells. So that is asexual reproduction. And there's even some animals that are macroscopic animals that do that. Most of them, though, are single-celled. Then the second form of reproduction is sexual reproduction, and that involves two parents that are genetically distinct from each other. Each of them is going to make gametes, and then they're going to send those gametes somewhere where they can meet, and they come together to form a diploid cell, a cell that's got two sets of chromosomes that then is genetically unique from both of the parents. Okay, so we end up with two organisms that are different from each other, and we make a third one that is different from the first two. Maybe some similarities, but definitely different. That occurs via meiosis, and that's what we're learning about today. And then two steps. Meiosis is where we make the gametes, and fertilization is where sperm meets egg. And that's where the third genetically distinct organism is going to be created. Anyway, I'm being very technical here about it, but clearly, even in the state of South Carolina, we've had some sexual education, and hopefully we can step into this without too much trouble. Okay, well, here's another slide with a lot of words on it, but this is really just kind of a explanation slide. There's not a lot of terminology here. And it has to do with a question that biologists like to like to address, because when students learn about this asexual and sexual reproduction, they're often left with the question of why. The way that I described asexual reproduction to you, it's easy. You just pop off a cell, it grows into a new organism. That seems like the way to go. It requires less time, less energy, and you can make tons of offspring from a multicellular organism. So that's what we have here. The question that we're dealing with is, why is sexual reproduction successful? Why would that even evolve? Because it requires more time, it requires more energy, and it creates fewer offspring. That doesn't seem like a success story. But the key is this last thing on my list. It generates genetic variation. That means that third organism, that offspring that is genetically distinct from the parents. And in fact, if those parents make a whole bunch of offspring, all the offspring are also genetically distinct from each other. So we're making a generation of organisms that are all slightly genetically different from each other. With asexual reproduction, that's not the case. They're all identical. With sexual reproduction, they're different, and that is why it is so successful, especially for eukaryotic organisms, large organisms that are mobile and might move from one environment to another. Because if you move into a new environment, then you need a different set of genes in order to be successful in that. And so sexual reproduction is what I would call hedging your bets. It's basically diversifying your portfolio. You are creating a bunch of investments that are all slightly different from each other in the hopes that one or two of them are going to find the environment that they will be successful in. Biology is about life, but it's also about death, because organisms, when they reproduce, most organisms that are reproduced, and I mean not just humans, but all organisms, most of them are bound for death before they get to reproductive age. And that means that, hedging your bets would require making diversity so that as many different designs make it to reproductive age as possible. I don't know if I've explained that, but this list down here says that it increases the probability of a small number of surviving offspring. Hedging your bets, diversifying your portfolio. I don't know if those metaphors help. Anyway, that's why sexual reproduction is so successful among larger eukaryotic organisms. Okay, so here we see the word meiosis, and it is underlined, and I've highlighted it, so that's important. Mitosis, meiosis. Some of you may have heard it pronounced meiosis, but that's not the way I was taught, and I tend to think mitosis, meiosis are easier to kind of remember. All you got to do is take the T out, and you've got the other one. So meiosis is the formation of gametes. What we know about gametes, because we learned a little bit about cells in the last lecture, is that sperm and eggs, gametes, for humans, have 23 chromosomes. We learned that. But all of the rest of the cells, all the regular cells, have 46. And the terminology that goes with that is that gametes are haploid cells. That means they have half as many chromosomes as all the other cells, and that the rest of the cells are diploid cells. So the process of meiosis is taking diploid cells and making them into haploid cells with only one set of chromosomes, so that those two cells can meet and form another diploid cell. And that's how the shuffling of all the genetic material comes about. So meiosis produces haploid cells, and then fertilization, bringing those haploid cells together, those are the gametes, turns them into diploid cells. And we call that first diploid cell a zygote, which you may have heard, which is basically a fertilized egg. All right, so the pictures that we have down here are showing some humans, which are animals, and some fungus and some plants. And these are all eukaryotic, sexually reproducing organisms. And so they all have phases of their lives which are making gametes, which are haploid, and then coming together to form diploid somatic organisms. But it's different for each one of those. And I want you to know that with humans, the humans that you see, the bodies, the parts of the human that you see most of the time, those are all diploid cells. Whereas if you looked at a mushroom or a mold growing on your bread, if you look over here, most of it is in this gray area. That's the haploid region. What you're looking at is haploid tissue. They become diploid just for a little while to reproduce. So it's kind of just the opposite. And plants kind of split the difference. Part of their lives are spent diploid and part of their lives are spent haploid. The diploid parts are usually too small for us to see, but they're there most of the time. So we call that alternation of generations. But this is not a botany class. It's a human biology class. So we're focused over here on the left, and let's take a look at that. In the blue, that's where we're diploid. We have some people, and they have some special organs that are making gametes, the testis in the man and the ovary in the woman. And we get a fertilized egg, which then becomes a zygote. And this is a sperm right here, and this is the egg right here. And then they're going to meet, and that's fertilization, and that forms a zygote, which then grows into another person. Again, genetically different. So that's what we're looking at in meiosis, is how do we make these haploid cells? And clearly this might involve recalling a little bit of that sex education that hopefully you had some of. Okay, so now here we have some details, and we're going to be talking here about meiosis. And we want to distinguish it from mitosis, and one of the first things that we notice right away is that there are going to be two cell divisions. So one cell is going to split into two, and each of those two cells is going to each split into two. That's going to make 4 cells altogether. That's really different, and it is in the first cell division, which we call meiosis 1. That is where we're going to be going from a diploid cell to 2 haploid cells. So that's where the really major difference is. It's in meiosis 1. So it says here that first we're going to replicate the DNA, and we saw that in the cell cycle back in mitosis. But then in mitosis, the chromosomes lined up in the middle, and they all split in half, and so each of the daughter cells got 2 identical sets of chromosomes. Okay? In this case, that's not going to happen. They don't all line up right on the middle. They line up kind of on either side of the middle, and then they do some swapping of parts. It's a strange, complicated process. I'm just going to call it chromosome shuffling for now. All right, so everything gets shuffled. It's kind of like you have a deck of cards, and you've got 4 sets of the numbers in the cards, and then you're going to shuffle them up before you separate them into 2 separate half decks. I don't know if that helps or not, but we're going to do something really different in meiosis 1. And it involves rearranging the chromosomes so that we make 2 haploid cells in that process, but they're already genetically different from each other. Right after meiosis 1, right after the first division, they're already different. And that's what we're after, is that genetic variability. So meiosis 2 involves each of these cells dividing into 2 cells. And that's a lot more like mitosis. It's really quite a bit like mitosis, except that in that shuffling of chromosomes, the sister chromatids that we're dealing with in each of these cells are not the same as each other anymore. So when these 2 cells divide into 2 daughter cells of their own, I wonder if they call those granddaughter cells, because this would be from parent to daughter cells, and then these would be granddaughter cells. I've never heard that. But anyway, each of these is also genetically distinct from its parent, and so it's still haploid cells, but all 4 of the cells produced by meiosis are genetically different from each other. Remember, we're hedging our bets. We're trying to make diversity to make sure that some of the offspring survive. Okay? The other thing that's in here, I think that's important, is we've been talking a lot about eggs and sperm, but we don't always use those terms. Sometimes we use the word spores. Sometimes you might hear people talking about pollen grains, which actually have cells inside that are going to become sperm. Those are all involved in this process of meiosis, making haploid cells. So for animals, though, we generally use the terms eggs and sperm. And then, obviously, following meiosis, gametes are released, and that's done in lots of different ways, but somehow egg needs to meet sperm, and that's going to bring them together to form a new diploid zygote. And we all have an idea of how that happens in humans, at least I hope. All right, so here we are with a big terminology slide here, but there's good news. First of all, this is only meiosis 1, but the good news is that the terms here are the same as the ones we've already learned for mitosis, except that we need to put a little bit of extra information on them. So instead of the 5 stages that we learned, we're now going to talk about prophase 1, prometaphase 1, metaphase 1, anaphase 1, and telophase 1. And as you can imagine, when we get to meiosis 2, the second cell division, we're going to see that again with the 2's on the end. And of course, we have interphase, but we're not going to divide that into interphase 1 and 2. We just call it interphase. So that's the same. In fact, I've written here, let me just get my other color, that is just like mitosis. So we go to prophase 1, which is where the really important shuffling goes on. In fact, we have a different word for it. We call it recombination. Basically, the homologous chromosomes, the ones that are one from mom and one from dad, they find each other and they line up and they reach out and trade some parts. It's kind of odd. I'll show you some pictures. So we call it recombination, or sometimes it's called crossing over, because that's kind of the way I'm with these arms of the chromosomes reaching out and swapping parts. So that happens in prophase 1, the reshuffling. Okay. Then in prometaphase 1, we're going to line up at the metaphase plate, but it's not quite the same. It's not like every chromosome is lined up right on the plate. What they do is they line up on opposite sides of the plate. So first there's the reshuffling, then there's a slightly different lining up, and they slide apart from each other so that each new cell is going to get a genetically distinct set of chromosomes. So this one is very different from what we know because of that shuffling, and this one is different because they line up in a slightly different way. That's really important. Beyond that, it starts to look an awful lot like mitosis. So we've identified the 2 parts that are really different. Metaphase 1 is really just beginning to slide apart. The spindle fibers reach out, grab things, and pull them apart. Anaphase, we can see the chromosomes being separate, and then in telophase, they start to develop, their separate cellular parts, and it's a lot like mitosis. So look to prophase 1 and prometaphase 1 for how things are a little bit different in this stage. Remember, we're only ending up with a set of haploid chromosomes in each of those daughter cells. Okay, so here's the pictures I promised, and they look similar to some that we saw before. We're going to start. I didn't leave myself very much room for myself here, so I'm in the nucleus there because nothing is different in interphase. We've already seen that. Things start to get really different in prophase 1, and this is where the crossing over or recombination takes place. And what we have here is red and blue are representing chromosomes from mom and dad, basically, and they are they're coming together, and you can see that the arms are reaching out and swapping parts, so that when they line up at the plate, and they're not going to line up right on the plate, they're going to line up on opposite sides of the plate. That's what we have. That's what we're showing here in metaphase. When they do that and then slide apart in anaphase 1, the result is that what's happening is the set of chromosomes that each of the new cells is going to get are already really different from each other, and you can see that in the red and blue parts. They're just using colors to represent genetic differences in this case. Okay, so the steps are interphase is the same. Prophase 1 is where recombination and crossing over take place. Those are 2 different words for the same thing. And then the lining up during prometaphase and metaphase means that they're on opposite sides of the metaphase plate, and by the time they slide apart, they are genetically distinct. And that information is down below as well. All right, so here's the slide with all the steps of meiosis 2, and the great news here is that there's hardly anything that's different at all from mitosis, which we've already learned. One of the differences is going to happen actually in interphase. Remember, in interphase we had the G1, the S, and the G2 phases within that, and S is where we made a copy of all the DNA. We're not going to do that. There is no copying of the DNA. So it says no replication during second interphase or interkinesis. So interphase 2, if you want to call it that, it's called interkinesis, has no S phase. It can be checked for mistakes, but there is no S phase. So we're going to, in this case, in meiosis 2, each of the daughter cells is a haploid cell, and it is going to produce 2 haploid cells. Because there was no copying, we're just going to pop apart the sister chromatids that are on each of those chromosomes. Okay, so no replication, and then prophase 1, prophase 2, prometaphase 2, metaphase 2, anaphase 2, and telophase 2 are pretty much just like mitosis. All right. In fact, they're all going to line up on the plate the way they didn't do in meiosis 1, and the centromeres are going to pop and the sister chromatids are going to slide apart, just like in mitosis. Boom. All right. So again, not a lot of room for me, but here's the diagram that's showing meiosis 2. We're actually starting in telophase 1 and cytokinesis from after meiosis 1. And then I guess this first set of blue arrows here represent interkinesis or interphase 2 leading into prophase 2. And what you can see is that we have our chromosomes that have already done in meiosis 1, they've already done the shuffling, the swapping, crossing over, and the rest of this looks a lot like 2 sets of mitosis going on. What happens is the chromosomes all line up, not on opposite sides of the plate, but right on the plate, and then the spindle fibers come out, grab the chromosomes, and the sister chromatids pop at the centromere, and so we get a separate copy of chromosomes, but you see that they're slightly unique from each other because of that crossing over. And as a result, we have 4 haploid final daughter cells. I think I'm going to start calling these the granddaughter cells because we made daughter cells in meiosis 1, and now they're making daughter cells, and we all know that daughters of daughters are granddaughters. Okay? But anyway, this looks very familiar, very similar to mitosis. Okay, so this is a slide about randomness, and there's a picture of shuffling some cards because that's why we shuffle cards, is to produce some randomness in the arrangement of the cards in a deck. This is important because we indicated that sexual reproduction was about producing genetic variation in sperm and eggs, so that they can come together and then produce offspring that are distinct from their parents and distinct from each other. And so we kind of have to have a sense of where that randomness comes from, and we've seen some of it, but this is a summary that brings it all together and says, look at all this randomness. So it says here that there are 4 separate events in addition to mutation, which sometimes happens when genes change all by themselves. That's another story. But 4 separate events that contribute to genetic reshuffling. The first is what we call chance synapsis and crossing over during meiosis 1. That is prophase 1. That is where the chromosomes find each other and reach out and swap parts. That's the reshuffling that I talked about. So that's the first one. The second one is when they line up at the plate, okay, they can line up this way or they can line up this way, and that's going to determine which side, which gamete each set of chromosomes gets put into. And let me just tell you that it's different for every one of the chromosomes. Remember that there are 23 different chromosomes, 46 altogether. So for each one of those chromosomes, they could line up this way or they could line up this way. So that means that the final products are going to be shuffled once again. And then we're going to do the same thing in meiosis 2, except it's not lining up on opposite sides of the plate. It is one chromosome, but it could line up this way, or it could line up this way. Okay, so the pinky could be going to this cell, or it could be going over to this cell. And that's another, and each of the chromosomes is independent in terms of the way that they do that. So again, another opportunity for randomization. And then finally, the last one is that not all of the sperm and eggs are released at the same time, and which sperm meets which egg is going to have a lot of randomness in it, and so chance fertilization in mixtures of gametes. So a lot of randomness going into the genetic complement of chromosomes that a new individual offspring is going to have. And we know that, all right? Kids don't look alike. There's all kinds of reshuffling going on in this whole process. But we just want to make sure we acknowledge those are the 4 points at which the randomization happens. Okay, so here's a little bit more detail, just some diagrams of what I was talking about. Remember, there are 4 steps of generating some randomness, 4 ways of doing it, and they're all happening in this process of meiosis. The first one is happening in prophase 1, and we call it recombination. This is where homologous chromosomes, one from mom and one from dad, are going to line up next to each other, and then they'll reach out and do some trading of parts. That's called crossing over or recombination, and the result is that we get recombinant chromosomes where certain parts are not going to end up in the same cell with each other again when they were in the same cell originally. That is the first kind of randomness. The second kind is going to be happening in metaphase 1, and that has to do with which chromosomes line up on which side of the metaphase plate. In the upper example, we have well, the metaphase plate is actually vertical. So we have the blue chromosomes on one side and the red chromosomes on the other side. And then down below we have a blue and a red of each type of chromosome. So that's a different arrangement. We call that independent assortment. So recombination in prophase 1, independent assortment in metaphase 1. And then there's another level of independent assortment because we do it again in metaphase 2. We're not showing it here because there's no recombination that's visible in this. But remember, in metaphase 2, the chromosome lines up. But it could be in this orientation or it could be in this orientation. So depending on which way we go, the chromosomes are going to be popping apart and sliding into different cells. So 2 different names here for this. Recombination is the crossing over, and then independent assortment is about the way they line up during metaphase. And then the last source of randomness is what we call chance fertilization, which sperm meets which egg. But it's a little bit more than that because all the eggs are different and not every egg gets released all at once. So which egg is getting released? And then, of course, all the sperm are different and which sperm are getting released. And then there's which sperm is going to meet the egg, and that all of that is adding some randomness. And so we draw pictures like this, but this is a horrible, horrible picture because it is the worst kind of anthropomorphism that we can exercise in biology. The reason it's horrible is because all the sperm are swimming toward the egg, and it implies, and I think this is sexist, it implies that the sperm are the ones who are racing to do the fertilization, which sounds very human social systems. In reality, sperm have no brain, they have no sensory system, they swim randomly. The fact that they're all swimming toward the egg is just ridiculous. They swim in random directions, and it's actually the egg which determines which sperm gets to do the fertilization. So there's no race, there's no look who's talking, having a big fight to see who's going to get the girl first. This is just foolish, and I'm sorry that I even showed this picture. But, you know, it shows lots of sperm there. Okay, last slide, and it's a summary slide. It has on the left mitosis and on the right meiosis so that we can talk about some of the differences, and so I'm just about done. We start with the same basic type of cell. It's a diploid parent cell, and in this case we've got 4 chromosomes, 2 copies of each one. That's why there's 2 blue ones and 2 red ones. And we're going through prophase and metaphase and anaphase and telophase in mitosis to produce 2 identical copies of daughter cells, and each one of these daughter cells looks exactly like the parent cell. So we got all the names of the parts, but we're essentially doing asexual reproduction and cloning the original cell. However, over in meiosis, we're not doing the same thing. Instead of the normal lining up and dividing, these chromosomes are going to find each other, homologous chromosomes are going to find each other and do some swapping, some crossing over. And so we end up with 4 completely different granddaughter cells. And remember that these are haploid. Each one of them only has 1 copy of each of the types of chromosomes. So the types of chromosomes are the small chromosomes and the large chromosomes, and there's 2 copies of each one of those. Down here, each of these granddaughter cells only has 1 small one and 1 large one, and they're holding parts of chromosomes that they met earlier in the process, so they're all genetically unique, distinct. And that's essentially the difference between mitosis and meiosis, sexual reproduction involving creating genetic heterogeneity or genetic variation. All right, that's that. Hope I didn't talk too much about sex and make you uncomfortable, but this is biology class. And I'll see you in class, and stay safe.